This is the first of today's lecture worksheets, and we are looking at, what are we looking at? 12.3. Okay, so the 12.3 lecture, this is the functional groups on these modern drugs that are analgesics. And we'll see these structures look a little more, I guess, more complicated than the normal inorganic molecules we've seen. But because we understand now line angle drawings and functional groups, we know what we're looking for. So in looking for functional groups, a functional group is going to usually have atoms that are not carbons and hydrogens, or at least not only carbons and hydrogens. You'll throw in an oxygen in there with a carbon or a nitrogen in there. So we looked at the 7 functional groups, the alcohol, and you want to be able to recognize these by structure. So briefly above them, the condensed structural formulas. Alcohol is an OH group, and ether is an oxygen, and aldehyde is a carbon, double bond oxygen, hydrogen. A ketone is a carbon, double bond oxygen. Carboxylic acid is a carbon, double bond oxygen, single bond OH. And ester is a carbon, double bond oxygen, single bond oxygen. And then an amine is any nitrogen. But it's really the simplest form of the amine is just a nitrogen attached to the carbons and then 2 hydrogens. Okay, so notice that these are groups of atoms that will give a function to a molecule that's just a bunch of carbons that normally wouldn't have any function. As I mentioned in the last lecture, when you have a hydrocarbon, all it does is it really just burns. And so you don't have that going on in biochemistry. You don't only have burning going on, you have all kinds of specific biochemical reactions going on that equal life. And it's thanks to these types of functional groups that allow for reactions that aren't just combustion reactions, that aren't just burning. Okay, so these are the functional groups, and here's one. This is the carboxylic acid structure. Here's the condensed structure. And then you also want to recognize the name endings. When you are looking at these, let's say, ingredients, this is an assignment from lab, is when you look at a lotion bottle or shampoo bottle, you can see the name endings will help elucidate what these molecules are all about. And they're not just a bunch of, foreign names or toxic drugs. They are really just putting together the structures, parts, and then naming those parts by being branches or having functional groups in them. Okay, so this molecule is called epinephrine, this molecule right here. And from the name ending, so that's, what's at the right side of the name, what functional group is present. So this is the name ending is INE. So when you have an INE name ending, that means you're looking at an amine. And so there must be a nitrogen in there. There must be a nitrogen or some NH2 group in there. Okay, so let's look at the structure and see if we can find it. Well, yep, here it is. So anytime you have a nitrogen bonded to anything else, it's any nitrogen. So this is a nitrogen with 1 hydrogen and bonded to 2 other carbons. This is your amine group. Okay, so anytime you just are looking at carbons and hydrogens, those are not functional groups, right? But this one, anytime you have a different atom, so this is an OH group, that's an alcohol. Here's another OH group, this is also an alcohol. And here's another OH group, that's also an alcohol. This is something that it does not have different atoms, but is a common structure. It's the benzene ring. It is the 6-carbon ring with 3 double bonds. Okay, and so this is on the epinephrine, and you can see this epinephrine has a lot of atoms in there, but the most important part, and the reason why it has the INE name ending, actually let me move this down a little bit, is that this INE name ending, what's highlighted is this amine. The amine is what makes this epinephrine, which is part of the fight or flight response in your human body. Adrenaline, adrenaline, epinephrine, they both end in ine, and that nitrogen is what allows for this to have a function that leads to that physiological response of fight or flight. So these are also these analgesics that we just talked about, aspirin, but also analgesics are ibuprofen and acetaminophen. So you might have heard of ibuprofen, the trade name is Advil, or acetaminophen, trade name is Tylenol. Well, so these are all analgesics, which are pain relievers. Okay, and so when you take one of these drugs, it initiates a pathway that cuts down pain or the sensation of pain in your brain. So let's label the functional groups in these analgesics. So all of them, what they all have in common is they all have a benzene ring. So let's label that benzene ring as the starting point. And so here's a benzene ring on each of these. Okay, so they all have a very similar structure that begins with that benzene ring, that thing. Okay, and so that's really the scaffold. And then what is also in common is, let's see, this aspirin. This is a functional group that is the carboxylic acid. It is the COOH group, but that's the structure all filled out. And then also here, this is the carbon with two oxygens close by. That's called the ester group. And that's a COO. And that's in the middle of, a carbon chain. So there's carbons on either side of the oxygen, and the other side of the ester, the carbon with the double bond oxygen. So you can see that on this benzene ring, there are two functional groups nearby. And this must mean that it's like a puzzle piece that fits into a receptor that then initiates the physiological response. So for the ibuprofen and acetaminophen, they're actually better versions of aspirin as far as pain relief and fever reducing, which is why we tend to take these instead. I can't think of the last time I took aspirin, in fact. Instead, I'm reaching for the Advil or the Tylenol. Okay, so, and why? It's because these structures are similar but slightly better, which means they have a slightly better fit, and then a slightly better physiological response. So the slightly better fit is because notice that these two groups are across from each other as opposed to next to each other on the benzene ring. And so what's needed is that starting point, but you also need to have, looks like, this same functional group, which is a carboxylic acid present. So that carboxylic acid is also on the ibuprofen. And there's basically the oxygens have these extra lone pair electrons that can initiate a reaction with those electrons. Starting point, though, is the docking, the fitting, the structure needs to fit with the starting point of the benzene ring, and then another structure on the other side of it, it looks like. So acetaminophen, Tylenol also has the same structure with the benzene ring and then two groups on either side. But the functional groups are slightly different. So when you have an OH group, that's an alcohol on one side. And then there's also, this is CO, that's our ketone group. And then anytime you have a nitrogen, that's an amine group. Okay, so you don't have to have the exact same groups, which is why this is not the exact same drug. Ibuprofen and acetaminophen have the same overall functions, but because they have slightly different functional groups, they're considered different drugs, right? So they're also, in addition to pain relief, they are antipyretic, which is fever reducing. And it's also why when someone has a high fever, it's recommended to take these two drugs in opposition. Like you can take ibuprofen, and then if you still have a fever, then you can take Tylenol, because it's not taking the same drug. So these drugs are also anti-inflammatory. And so this reduces inflammation. And what this points to is that these are different physiological responses, being pain relief, fever reducing, and reducing the inflammation response. So some of this is activating pathways going up to your brain, like pain relief and fever reducing, which is a different area of your brain, lowering your body temperature. And meanwhile, inflammation is locally, so in your joints. And so there are different receptors throughout the body that are affected by ibuprofen and or acetaminophen or aspirin. But it has to do with the starting point, which is the structure, and then having the functional groups initiates a series of reactions that are the physiological pathway. Okay. So when you have a pharmacophore, that's the basic structure, the most fundamental part of a drug that is needed to activate and produce a physiological response. So the drug receptor binding process that happens in a body, one example, it's called the lock and key model, where you have a receptor in the body that needs to be unlocked, basically. Somewhere in your body is a receptor. We saw that with how COVID docks into certain cells in your body, like lung cells or kidney cells. It's called an ACE receptor, and it is in those cells. It's not in all cells of the body. And so there are receptors spread throughout the body in different organs and different cells. And so what happens is that receptor has a shape, and in order to unlock it, in order to have a response, a physiological response, you need the right shape. Just like a lock needs the right shape of a key, because not just any key will open your front door, but a specific shape of the key will open your own front door compared to your neighbor's front door across the street. Okay, so which key will fit this lock is this one. Okay, and it has to have the right shape. And so this is the right shape. This has the can be a drug. This is your drug, or it was the COVID spike protein that fit into the receptor. But the key, once it fits into the lock, will then unlock the physiological response. Okay, just like when you fit the key into the front door lock, then it opens the door and you're in. Okay, and so when you have the key, that's the drug that unlocks the receptor in your body. And the structure needs to have also the ability to initiate the response. So we saw that with the analgesics, is that aspirin will fit into a receptor, probably the same receptor as the ibuprofen and the acetaminophen. But because the structure is slightly different, you can have a better fit. And so you need to have a specific structure that you really understand, that is the core of it, the pharmacophore. That's the arrangement of atoms that are absolutely required. You can have other stuff in there, but what you absolutely need at the very base is this. And you might have extras in there, but it has to be able to fit. Okay, so it fits into the active site of the receptor. So the active site of this receptor here is that shape, right? And so it's the shape, and also need to have the functional groups in the right positions to initiate that response. So this is the pharmacophore of opioids. So the opioid drugs have complicated structures. There's a lot more atoms in there, but this is the very fundamental foundation of the structure that's needed that binds into a receptor in the brain. So this is the activation of pathways in the brain that lead to the feel-good response. So the receptor in the brain that's targeted is called the mu receptor. And the structural features of this pharmacophore, so this is the very basic structure. There's more to it though, is it has a benzene ring. And then kind of funny is at an angle, but an angle coming out of the paper. That's why this is written kind of funny like that. It's an angle coming out of the paper or above the plane of the paper. You have this. Now, do you recognize that? That's an amine group anytime you have a nitrogen in there. So what you need literally is a benzene ring with coming out of it at a 90-degree angle in 3 dimensions is a nitrogen. Okay. And so this is where scientists then took off, and synthetic drugs are variants of this starting shape where you have a benzene ring and then a nitrogen above it at a 90-degree angle. So this is, these are other opioid drugs, morphine, Demerol. Morphine originates from the opium, but you can make it in a lab. So that's synthetic, right? Like you have synthetic polymers. And notice that all of these are the names of opioids. They all end in INE: morphine, codeine, heroin. They took the E off there. OxyContin, Vicodin. These are the structures have this pharmacophore, and they also are tweaked. They're slightly different so that they're allowed to have different drug names. And they are not exactly the same as far as the atoms, but they have the same base structure, so they initiate the same reactions. Okay, so these are the trade names, Demerol and Percocet. So they they probably do have the amine in there, but they have the different, they have the unofficial, the official trade names. So morphine has this pharmacophore structure with the benzene ring, and then a nitrogen group at this angle above it. So that's the amine group. And then this Demerol, also a pain reliever to a much greater degree than the pain relievers that we saw with the common analgesics, with the ibuprofen and the acetaminophen. Scientists really figured out how to take advantage of the pain relief, and created something called, created the OxyContin drug, which has led to the drug abuse problems, some of the drug abuse problems that we see now. Okay, and so that's the benzene ring with, at a 90-degree angle above it, the amine functional group. So this is complicated, it's fraught. Modern drug design, we need drugs to be developed, but scientists went a little crazy coming up with more versions. I'll scroll back down, sorry. More versions of these painkillers that originated from natural painkillers. And now we have drugs that are 1,000 times more addictive and also more effective, but also more addictive. So another important part about functional groups is not just that they give function to a molecule, but they also can make a drug soluble in water. So that has to do with the solubility, the ability of a compound to dissolve in water. And that becomes important in the human body because the human body is mostly made of water. So in general, the rule about solubility is similar things will attract together. Like dissolves like. So nonpolar goes with nonpolar. We saw that with the oil on your hand attracts to the fatty, greasy layer of COVID. Okay, so oil and oil. And the thing about water is opposite of oil. Water is polar. And so ionic and polar will not go with nonpolar. And in general, drugs tend to be nonpolar compounds. So for a drug to be effective in the body, it has to have some interactions with water. It has to have the ability to interact with water that we know has this bent shape. Here's the water molecule, and it has the partial negative end on the oxygen and the partial positive ends on the hydrogens. Okay. And so how the water molecule interacts with any molecule is that it is first attracted to the other water molecule, or from a water molecule to the other molecule, whether it's an ion or a polar compound, it's attracted to the partial positive and partial negative, or the full positive and full negative of whatever else is around. So water can be attracted to, this is a polar compound here. This has a carbon end, but when you attach an OH group, this is an alcohol group on there. When you attach the OH group onto the carbon, this makes this entire molecule polar because the oxygen-hydrogen bond is a polar bond. And remember how you determine a polar bond by taking the electronegativity values of the two atoms, in this case, oxygen and hydrogen. Those values you subtract, and if it's between 0.5 and 1.9, we count it as a polar bond. And that means the electrons are more, they're more attracted to spend more time on the oxygen, and that leads to a partial negative, and less time on the hydrogen in the bond, and that leads to a partial positive. So the water molecule, though, will then attract to this polar part of this molecule with attractions that are called intermolecular forces. Remember that, where you have the hydrogen, it has its partial positive, and it is the hydrogen on the water molecule. So here's a different water molecule. And here's even another different water molecule. The partial negative on an oxygen will attract to the partial positive of this molecule that has the alcohol portion on it. So those are attractions that are called hydrogen bonds. And the hydrogen bond, remember, is when it's one of those misnomer things. Remember, it's when you have an intermolecular attraction from one molecule to another molecule, where on one molecule, it's a partial positive hydrogen, and on the other molecule, it's either a partial negative, and it can be an oxygen, nitrogen, or fluorine. So this is a hydrogen bond when you have a partial positive hydrogen, that's this, attracted to a partial negative on another molecule, and it can be an oxygen, nitrogen, or fluorine. So here's another example of a functional group on this molecule. This is adding the entire functional group. You recognize COOH, that's carboxylic acid. But it has the OH bond. So the OH bond is a polar bond in there. And that polar bond, as part of the carboxylic acid right here, gives you a partial negative and a partial positive. So the attraction of water would be that this is the partial positive can be attracted to the partial negative of a water molecule. So the water molecule has the oxygen that has the partial negative, or this also has a carbon oxygen bond that is also, when you do the electronegativity calculation, is a polar bond as well. So that means that there's the oxygen has the electrons more of the time, and there's a partial negative. Okay, and so this would be attracted to the partial positive of a water molecule, which is the hydrogen end. Okay, and so those are, again, hydrogen bonds, because hydrogen is the partial positive. Okay, now most drugs are organic compounds, which means they are mostly nonpolar. They're just a bunch of carbons and hydrogens, and that calculates out to nonpolar bonds. But once you add in a functional group like an alcohol or an amine or that carboxylic acid, that will improve the solubility, the ability of the molecule to attract water molecules, and then the water molecules will surround and then dissolve. So for these molecules, these are examples that we saw in the video of vitamins. So you have vitamin A versus vitamin C. So a non-polar portion, that's the hydrocarbon part. So what's the hydrocarbon part of this vitamin A? Well, it's all of this that has just carbons and hydrogens. And what we recognize from the line angle drawing is it's all that zigzag part, and then some of the carbons are drawn in there. But there's the polar portion, which is just that OH bond. Now that is not enough to make this water soluble. So this is called lipid soluble, or it's another way to say fat soluble, which is another way to say that it is a non-polar molecule. Okay. Even though it has a polar portion, for the most part is a non-polar molecule, and it will only be attracted to other non-polar compounds like fats. And so this vitamin A is not only attracted to, but is stored in fat. So that's an example of those fat soluble vitamins. They're stored in fat, and you don't need them every day. That's vitamin A, D, E, and K. Versus vitamin C, which is listed as a water soluble vitamin. So what does that mean? It means that because it has these polar groups, the alcohol groups, those OH groups. Plus having any oxygens usually makes it polar as well. Having a lot of polar groups makes this an overall polar molecule, where the electrons are not distributed evenly. Okay, so you'll have partial positive and partial negative in there, and that allows for interactions with the water molecule, making this water soluble. So vitamin C is an example of a vitamin that you need to take every day because you're excreting it every day. It is not stored. And so, as I mentioned in the video, vitamin C, if you don't take it every day, you'll end up with a disease called scurvy. So vitamin C you can naturally get from foods every day. It was only in the extreme case of traveling across the Atlantic and you don't get enough of your all different kinds of foods when you're on a boat for months on end. So one way to improve solubility is to add any functional groups that are polar, like OH, the alcohol, or the amine group, or this carboxylic acid. Another way to improve solubility is to add an ionic end. Now we saw that with the soap molecule. So the soap molecule has the nonpolar tail and the ionic end, and that's added through the saponification reaction. So here's a process that is called, that turns pseudoephedrine. So pseudoephedrine is a common antihistamine, but it's actually converted a little bit into what's called pseudoephedrine hydrochloride, where there's an ionic end added to it. And it's a reaction that is used to improve the solubility, to allow that antihistamine drug to dissolve in the body and have an effect. And so this is where there's the ionic end. So the ionic end is a positive charge and a negative charge. And it's because of this reaction that this is the actual end result drug is pseudoephedrine hydrochloride is what you see on the label. Okay, and so all of this part here, this part makes it nonpolar, but adding the ionic end makes it polar or attracted to water. So you could have this negative attracted to the partial positive of water, like that. Or this positive can be attracted to the partial negative of a water molecule, like that. Okay, and so once the water molecules are attracted, then they can get in there and surround, and that will allow for this dissolving in the body. Process of dissolving starts with the surrounding. Okay,